Okay, we are going to start. Dear participants, my name is Maria Rodriguez, and I'm Project Manager for the EuroBloodNet. And it is my great pleasure to welcome you on behalf of the EuroBloodNet to the first webinar of the topic on focus on constitutional thrombocytopenia. This series is an accredited European online educational program targeting health professionals organized by the EuroBloodNet and in partnership with the CRPP, the French Reference Center for Inherited Platelet Disorders, and the Filière MHEMO, the French Constitutional Bleeding Disorders Network. The program consists of a cycle of 15 webinars developed in collaboration with 3 experts, Professor Marie-Christine Alessi and Dr. Paul Saultier from Assistance Publique-Hôpitaux in Marseille, member of the EuroBloodNet, and Dr. Mathieu Fiore from CHU Bordeaux. The main objective of this program is to disseminate up-to-date knowledge among interested hematologists, internists, pediatricians, or other healthcare providers to give visibility to cutting-edge advances in the field of constitutional thrombocytopenia. Before starting the session, I'm going to provide you with some final health rules. The session is recorded and will be uploaded in the EuroBloodNet e-learning environment. So if you do not wish to appear or being recognized, please turn your camera off or change your name in the platform. Also, please be kindly informed that your microphone is muted. If you have any questions about the presentation, please write them in the chat. They will be addressed during the Q&A session at the end of the webinar. You can find the chat at the bottom of the screen at the cloud symbol. As said, this program is accredited by continuing medical education points issued from the European Body of Accreditation in Hematology. You can request one credit per follow session or 15 credits if you follow all the program. How to request them? Please follow those 3 rules. Number 1, write your name and surname in the Zoom platform. Number 2, then in the chat, I will add a link redirect to an online form to fulfill in order to track your presence. You only need to insert name, surname, and email. And number 3, at the end of the webinar, you are going to receive a satisfactory survey consisting of 3 simple and quick questions. Please answer to it. So as said, today is the first webinar of this new series. Please join me in welcoming Professor Hana Raslova, who will lead this lecture on the pathogenesis of inherited thrombocytopenia. Professor Hana Raslova is co-leader of the INSERM team, from stem cell to megakaryocyte, located at the Institute Gustave Roussy in Villejuif, France. She has a long-term expertise in normal and pathological megakaryopoiesis. Her research is focused on the regulation of megakaryocyte ploidization and differentiation, and on the pathophysiological mechanisms of rare inherited thrombocytopenia, with and without predisposition to acute myelogenous leukemia. Professor Hana Raslova, the floor is yours.
So thank you very much for this nice introduction. So I am very happy to present you today the pathophysiological mechanism of some inherited thrombocytopenias. I have nothing to declare. Okay, so sorry. So the objectives of my webinar today are to explain you what are the different types of inherited thrombocytopenia, what is the purpose of a better understanding of the regulation of normal megakaryopoiesis, and why and how we can study the pathophysiological mechanism of inherited thrombocytopenias. So I will start with a small overview of the megakaryopoiesis. As you know, the megakaryopoiesis is a process that starts by the amplification of the megakaryocyte progenitors by classical mitosis that will then switch to endomitosis, leading to the increase in the DNA level. The megakaryocyte become polyploid, and then the maturation of cytoplasm with the biogenesis of granules and the development of a system of demarcation membranes takes place. At the end of maturation, the platelets are released into the blood circulation. In the in vitro cultures, there is a step when the demarcation membrane system will generate a network of proplatelets that are fragmented into the platelets. The whole process is regulated by different extrinsic factors, such as extracellular matrices and different cytokines, the most important being the thrombopoietin, and also by intrinsic factors, such as transcription factors. So the defects at any stage of the megakaryocyte differentiation will lead to the deregulation of megakaryopoiesis and to a defect in platelet production. Since several years, many genes were described to be mutated in inherited thrombocytopenias. First, we can find the mutations in the TPO-MIPL signaling, regulating all the steps of megakaryocyte differentiation, but particularly this axis is important for the amplification of megakaryocyte progenitors. Then we have the mutations in the genes encoding transcription factors, regulating megakaryopoiesis, in the genes regulating the metabolism, the granule biogenesis, and the cytoskeleton reorganization, which is important for platelet formation. And also we can find the mutations in the genes in signaling, regulating these cytoskeleton regulations. And finally, we have also mutations in genes which are involved in the sialylation of platelets. So some of these thrombocytopenias are characterized by the presence of platelets with normal size, here in black color. Some of them by the presence of platelet with small size, here in red color. But the majority are macrothrombocytopenia, which are here in green. Some of these thrombocytopenias are syndromic, and three of them lead to a predisposition to myeloid or lymphoid hemopathies. So first, I will speak about the inherited thrombocytopenia with affected TPO-MIPL signaling. In this group, we have mutations in the MIPL gene, leading to the congenital amegakaryocytic thrombocytopenia. In the gene encoding thrombopoietin, here we have two different types: monoallelic mutations with mild thrombocytopenia and biallelic mutations with a severe CAMT. And finally, we have here also the ankyrin 26 related thrombocytopenia, also called thrombocytopenia 2 or THC2, with mutations in the regulatory region of the gene encoding for ankyrin 26. So first, something about the CAMT. In majority of cases, the CAMT is characterized by autosomal recessive inheritance and mutations in MIPL gene. CAMT is a severe thrombocytopenia leading to the bone marrow failure disorder. Two groups were identified: type 1 with a complete MIPL loss of function and early progression to BMF, and type 2 with a residual receptor function and delayed development of BMF. Mutations in MIPL are localized through the whole gene, and here in green are indicated the less severe mutations. So here are some examples of MIPL mutations. For example, the mutation at position 635 will lead to the loss of function and MIPL with impaired stability, but this mutant can be stabilized by inhibitors preventing degradation. Then we have mutants affecting arginine at position 102, leading to the trafficking defect and poor surface expression of MIPL. And finally, we have the mutant at position 104, which will prevent the TPO binding. So recently, this German group has shown that the nonsense frame shift and the mutations leading to a complete loss of splice site will lead to the absence of MIPL at the cell surface of hematopoietic progenitors, while the missense mutations or compound heterozygous mutations will lead to the variable MIPL expression level as compared to healthy cells. So we have recently also reported the homozygous MIPL mutation at position 464 in a family with a consanguinity leading to a severe CAMT. To study the function of this mutant, we overexpress this variant in hematopoietic cell lines, and we have also studied the patient platelets and progenitors. We have shown that this mutation only partially reach the platelet level surface, the surface of platelets, and the same result was observed although also in the cell line after overexpression of this mutant. So by using the cell lines and the primary patient cells, we have shown that MIPL is not, this variant is not glycosylated and is retained in the endoplasmic reticulum, only weakly responds to TPO, but in the synergy of TPO with L-thrombopac slightly improved the signaling. As the chaperone of MIPL is calreticulin, because calreticulin is responsible for the retention of immature or misfolded MIPL and endoplasmatic reticulum, we hypothesized that the overexpression of mutant calreticulin could restore the MIPL level at the cell surface. However, this was not really the case. However, what we observed is that the expression of mutant calreticulin restored at least partially the signaling, but only in the presence of L-thrombopac, as you can see here. And this combination also leads to the restoration of proliferation of cells. So more importantly, we have shown that mutant calreticulin in combination with L-thrombopac restored the megakaryocyte differentiation of patient hematopoietic progenitors. So now something about the mutations in the gene encoding for TPO. So as you know, the mutations in the regulatory region of thrombopoietin or at the boundaries between the exons and introns leads rather to thrombocytosis, while the mutations in the coding region rather induce thrombocytopenia. This mutation can be monoallelic here in red or biallelic in green and can lead to different phenotypes. There is also one mutant here, which was found at the homozygous state in CAMT, but at heterozygous state in mild thrombocytopenia. A majority of mutants which are in receptor binding domain lead to a defect in the binding to MIPL, while the mutants in C-terminal affect the trafficking of thrombopoietin to the secretory compartment and therefore lead to a reduced TPO secretion. So interestingly, very recently, one Italian group has described a homozygous mutation in the regulatory region of TPO, leading not to thrombocytosis, but to thrombocytopenia. These mutations affect STAT4-H1 binding to the promoter region. The author showed that STAT4 and H1 positively regulate TPO expression, and their loss of binding to the promoter region of this mutant leads to the decreased expression of thrombopoietin. So the last thrombocytopenia affecting the signaling is ankyrin 26 related disease, or THC2. This thrombocytopenia is characterized by autosomal dominant transmission, normal mean platelet volume, moderate thrombocytopenia. It is not syndromic, and it is characterized to predispose to myeloid malignancies with low penetrance, and also by the mutations in the regulatory region of the gene encoding for ankyrin 26. So we have shown that these mutations lead to the loss of function of RANKS1 and FLI1 binding. RANKS1 and FLI1 are transcription factors regulating the normal megakaryopoiesis, and we have shown that they negatively regulate ankyrin 26 during normal megakaryopoiesis, and the loss of the binding to this region in patient cells will lead to the overexpression of ankyrin in megakaryocytes of patients and also in platelets of patients. So very recently, we have also shown that these mutations, the consequence of the overexpression of ankyrin 26 is that it will bind to MIPL, and this will induce the defect in the MIPL internalization, and this will lead to the hypersensitivity to TPO and L-thrombopac, and consequently to increased signaling and to a defect in proplatelet formation. So finally, we have shown that the inhibition of the increased signaling, and particularly of MAP kinase pathway, will restore the proplatelet formation, as you can see here. So then we have a group of thrombocytopenia with mutations in transcription factors. As I have already mentioned, different transcription factors regulate megakaryopoiesis, and the mutations in 8 of them were reported in inherited thrombocytopenia. So some of them affect only the megakaryocyte lineage, like FLI1 mutations, but other can affect also other hematopoietic compartments. For example, RANKS1 regulates also B and T cell differentiation, and also the number of hematopoietic progenitors. GATA1 can regulate also the erythropoiesis, so the mutations will lead also to the defect in the erythropoiesis. So germline mutations in RANKS1 gene are responsible for familial platelet disorder predisposing to acute myelogenous leukemia. And this disease is characterized by autosomal dominant transmission, by normal platelet size, moderate thrombocytopenia, and predisposition to myeloid malignancies. FLI1 deletion is responsible of Jacobsen syndrome, also called paritruso thrombocytopenia, which is characterized by autosomal dominant transmission, by slightly increased platelet size, moderate thrombocytopenia. And this disease is syndromic because other genes are deleted in this region. However, point mutations in FLI1 gene were also reported in FLI1-related thrombocytopenia, which is not syndromic. So GATA1 germline mutations are found in X-linked thrombocytopenia with dyserythropoietic anemia, characterized with a slightly increased platelet size, moderate to severe thrombocytopenia, platelet dysfunction, alpha granule deficiency, and the defect in erythropoiesis. So then mutations in ETV6 gene were described in ETV6-related thrombocytopenia, which is characterized by autosomal dominant transmission, normal platelet size, moderate to severe thrombocytopenia, and predisposition this time to lymphoid malignancies. Frameshift mutation in GFE1B gene generate truncated protein acting in dominant negative manner and leading to the deregulation of early steps of megakaryopoiesis with increased erythroid progenitors, resulting thus in thrombocytopenia associated with anisocytosis and poikilocytosis. So in the case of MECOM or MDS1 EV1 locus mutations linked to RUSAT1 or RUSAT2, the pathogenesis is not so clear. EV1 is involved in the maintenance of hematopoietic progenitors and in the regulation of MIPL expression, but the MIPL deregulation was not confirmed in all patients with MECOM mutations. So this disease is characterized with a with autosomal dominant transmission, is syndromic. It is characterized also by B-cell deficiency and by the progression to bone marrow aplasia. So finally, the missense mutations in IKZF5 gene were shown to lead to mild thrombocytopenia with autosomal dominant transmission, with normal platelet size, and reduced quantity of alpha granules. In this case, only the megakaryocyte lineage is affected. So now I will speak a little bit more about the RANKS1 mutation. So RANKS1 mutations linked to FPD-AML are localized in DNA binding domain or in the transactivation domain, but intra or intergenic deletions were also detected, as well as the amplification of the mutant allele. Two types of mutations were reported: the haploinsufficiency-like mutations, leading rather to thrombocytopenia alone, or dominant negative-like mutations with higher risk to develop myeloid malignancies. We and others have shown that RANKS1 regulates megakaryopoiesis at different stages. It regulates negatively the number of immature hematopoietic progenitors, but positively the number of megakaryocyte progenitors. It regulates both induction and arrest of endomitosis. It regulates also proplatelet formation and also the platelet functions, and this via direct regulation of different RANKS1 targets. Some of them are regulated positively, here in red, and some of them are regulated negatively, here in green. The deregulation of all these genes could contribute to the thrombocytopenia. One of them is ankyrin 26 that I already presented, and the second gene that we have found to be negatively regulated by RANKS1 is myosin heavy chain 10. So we have shown in our lab that MIH10 is well expressed at the first stages of megakaryocyte differentiation, but that its expression progressively decreases and is almost completely silenced in the mature megakaryocytes and in platelets. Interestingly, we have shown that this silencing is necessary for the switch from mitosis to endomitosis, and that it is achieved by negative regulation of MIH10 by RANKS1. These results were also confirmed in a knockout RANKS1 mice, showing an increased expression level of MIH10 in platelets. So this led us to hypothesize that MIH10 overexpression leading to a generation of hypoploid megakaryocytes could be at least partially responsible for a thrombocytopenia of FPD-AML patients. And we have confirmed that MIH10 is really expressed in small patient megakaryocytes, but not in polyploid control megakaryocytes. Finally, we have shown that inhibition of ATPase activity of myosin by using a chemical inhibitor blebistatin considerably increased the ploidy level of the patient cells. So more importantly, these results led us to develop a diagnostic test based on the expression of MIH10 in patient platelets. As shown here, MIH10 is well expressed in platelets of all FPD-AML patients, but not in control platelets. By screening the platelets of other thrombocytopenias, we have found MIH10 expression also in platelets of patients with Paris-Trousseau syndrome, which is not surprising because RANKS1 and FLI1 are in the same transcriptional complex that downregulates MIH10 expression during normal megakaryopoiesis. So we postulated that the presence of MIH10 in patient platelets could be indicative of the presence of RANKS1 or FLI1 genetic alterations. And this was confirmed by the work of Paul Saultier, showing by fax analysis that patients carrying two different FLI1 variants express MIH10 in platelets of these patients, confirming the functionality of these two variants. So coming back to the Jacobsen or Paris-Trousseau syndrome, which is a syndromic disorder characterized by a monoallelic deletion of FLI1 and by a presence of two different megakaryocyte populations in bone marrow, one normal and one small apoptotic. We have shown that during the normal megakaryopoiesis, there is a stage of maturation just before CD42 expression, at which only one allele will express FLI1, and then the biallelic expression will be restored. In patient megakaryocytes, if the conserved allele will express FLI1, normal megakaryocytes will be generated. But if the conserved allele doesn't express FLI1, the small megakaryocyte will be produced and will die by apoptosis. So now something about the GATA1 linked thrombocytopenia. So it exists two isoforms, GATA1 isoform, one translated from the first exon and the second from the second exon. So mutations in the second exon will lead only to a GATA1 short isoform, which is found in megakaryoblast leukemia. The mutations localized in the N-terminal zinc finger domain will lead to the macrothrombocytopenia, and depending on the loss of DNA binding or FOG1 binding, the patients will present with severe dyserythropoietic anemia with macrothrombocytopenia, or with macrothrombocytopenia with mild dyserythropoietic features, with macrothrombocytopenia with beta thalassemia, or by macrothrombocytopenia with congenital erythropoietic porphyria. Interestingly, the mutation that removes the stop codon will only lead to the thrombocytopenia, to the mild thrombocytopenia. So one of GATA1 targets, which is MBAL2, is responsible of gray platelet syndrome, which is also characterized by thrombocytopenia. So recently, two other variants were described in Marie-Christine Alessi's group. One in the N-terminal zinc finger domain, and the second in the C-terminal zinc finger domain. In both cases, the thrombocytopenia is characterized by normal platelet size. The first led to the severe thrombocytopenia with a mild dyserythropoiesis, and in the second case, the thrombocytopenia was initially absent and developed only over time. In both cases, the functionality of these variants was confirmed by a presence of MIH10 in platelets, and it was confirmed by different methods that GATA1 co-regulates MIH10 expression together with FLI1 and RANKS1 complex. So now I will present some thrombocytopenias with defects in the genes regulating the cytoskeleton reorganization and in the signaling pathways important for the cytoskeleton reorganization. So the cytoskeleton reorganization, important for proplatelet formation and platelet production, is regulated by GP1B, GP9, GP5 complex, which is the receptor of von Willebrand factor, by alpha-2b beta-3 complex, which is the receptor of fibrinogen, or by alpha-2b beta-1, which is a receptor of collagen, and also by small GTPases, such as RHO-A, CDC42, and RAC, which are downstream of these receptors. So RHO-A co-regulates actomyosin activity, actin stabilization and polymerization, and microtubule stabilization, through different effectors such cofilin and MLC through ROC, and by diaphanose-related formins, and more particularly the DAPH1. So the role of DAPH1 in cytoskeleton reorganization during proplatelet formation was for the first time described in 2014 in our lab, and the studies have shown that the inhibition of DAPH1 by shRNA lead to the decreased stress fiber formation and accelerated microtubule elongation, both resulting in the increased proplatelet formation. So DAPH1 could control proplatelet formation by regulating the balance between actomyosin complex and microtubule system. So 2 years later, the first variant in DAPH1 leading to macrothrombocytopenia was described. This variant is localized in diaphanose autoregulatory domain and generate a truncated protein. It was reported in 2 unrelated pedigrees with macrothrombocytopenia and sensorineural hearing loss. In patient platelets, an increased filamentous actin, here in red, and stable microtubules, here in green, were detected, suggesting that this variant leads to the constitutive activation of the DAPH1 that negatively regulates proplatelet formation. So then we have the mutations in the head motor domain or in the tail domain of myosin heavy chain 9 that induce the most common macrothrombocytopenia, also called MIH9 related disease. What has been shown is that the adhesion of patient megakaryocytes leads to the increase in stress fiber formation as compared to control megakaryocytes. And by traction force microscopy, it was shown that this leads to a strong increase in contractile forces in patient megakaryocytes as compared to controls. And this suggested that these mutants act not as haploinsufficiency-like mutants, but acquire a new function. So to investigate whether the myosin 2 hypercontractility contributed to a decrease in platelet formation in this disease, the megakaryocytes were treated with blebistatin that inhibits the myosin ATPase activity and by ROC kinase inhibitor, and it was shown that both these inhibitors improve the formation of the proplatelet formation by patient megakaryocytes. So the next one is the filaminopathy A, which is linked to the chromosome X. Its hemizygous loss is lethal, and the female patients with heterozygous mutations in FLNA gene present with multiple clinical manifestations, including macrothrombocytopenia. Our working hypothesis was that as random X inactivation occurs during embryogenesis, the megakaryocytes expressing Y-type allele will differentiate it correctly and produce normal platelets. And the megakaryocytes expressing deleted allele will present a defect in maturation and will produce macroplatelets. And this will lead to at the end to a mixture of normal platelets expressing filamin and macroplatelets without filamin A, as it was shown for patient platelets. So to confirm our hypothesis, we generated the isogenic IPS lines from 2 patients. As each IPS line comes only from one cell, we obtain the cells expressing only the Y-type allele and the cell lines expressing only the mutant allele. So by using this model, we have shown that megakaryocytes that don't express filamin A form after adhesion on fibrinogen increased stress fibers, suggesting an increase in actomyosin contractility, and they display a deep defect in the proplatelet formation. We have confirmed by different methods, not presented here, that this defect is, as before, linked to the deregulation of ROA pathway, and that the inhibition of ROA effector ROC decreases the stress fiber formation, and more importantly, restores at least partially the proplatelet formation, as you can see here. So overall, we have shown that in normal condition, the interaction between filamin A and alpha-2b beta-3 integrin is necessary to keep ROA at inactive state, and that in absence of filamin A, ROA is activated, and this leads to increased actomyosin contractility and to macrothrombocytopenia. So the mutations in the glycoprotein receptors also lead to macrothrombocytopenia. Between them, we can find Bernard-Soulier syndrome, characterized in a majority of cases by biallelic, homozygous, or compound heterozygous mutations in GP1BA, GP1BB, and GP9 genes. But in some rare cases, we can find also monoallelic mutation, the most common being the Bolzano mutation. We have recently developed the IPS model from a patient with a new, not yet described heterozygous mutation in GP1B alpha gene. And we have shown that this mutant doesn't affect the GP1B, GP9, GP5 complex, its expression at the cell surface. We have shown that it doesn't affect the megakaryocyte maturation, but it leads to a decreased affinity to von Willebrand factor, and it alters platelet generation through ROA activation. By using the 3D silk-based model developed in Alessandra Baldwini's lab, we have shown that the treatment of patient megakaryocytes with ROC inhibitor, that this treatment increases the number of platelets, and it also restores the platelet size. So the restoration of platelet formation by inhibiting ROA pathway was confirmed for another patient, and this time the primary patient megakaryocytes were ground inside the 3D system, and the inhibition of ROC was shown to lead to the increase in platelet count. We observed a 2.5-fold increase in the number of platelets generated by this system. But this is also to demonstrate that the use of such 3D bioreactor can be useful to predict the drug response. So the last macrothrombocytopenia that I will present is von Willebrand disease type 2B, characterized by gain-of-function mutation of von Willebrand factor that enhances its binding to platelet GP1B alpha. As shown by Alexandre Kosko in mouse model, this mutant leads to the actin disorganization megakaryocytes through upregulation of a limb kinase coffilin pathway downstream ROA. And he has also shown that the use of a limb kinase inhibitor and ROC inhibitor were able to rescue the actin turnover and the proplatelet formation, and also the platelet size. So to conclude, the majority of the mutations affecting the reorganization of actomyosin and microtubule cytoskeleton leads to the deregulation of the ROA and CDC42 balance. And almost all of them lead to ROA overactivation that will induce the generation of macroplatelets. So to summarize, to incriminate each new mutated gene as responsible for inherited thrombocytopenia, it is necessary to study its function in normal megakaryopoiesis. To study the study of pathophysiological mechanism leads to the identification of new biomarkers that could be used for the determination of the variant pathogenicity. Study of pathophysiological mechanism permits to identify new deregulated pathways that could be targeted. As an example, the ROA pathway seems to represent a good therapeutic target to treat macrothrombocytopenia. The use of a three-dimensional bioreactor is helpful in predicting the drug response and could be used in personalized medicine. So overall, a better knowledge of pathophysiological mechanism leads to improved patient care. So finally, I would like to thank all the collaborators from our lab and also from Kremlin-Bicêtre lab for this, everybody who collaborated with us in these experiments. And also thanks to all the clinicians participating to this work, and also my external collaborators from Italy and from Argentina. And thank you very much for your attention, and I will be happy to answer your question. Okay, so I think I will. So I have a question. I'm not sure that you can hear me, but I suppose yes.
Yes, Hana, we can hear you.
Okay, so I start with the question. So the first question from Jose Rivera: any hypotheses about mechanism causing also neutropenia and DAPH1 related disease? No, I'm sorry, but we don't study the neutropenia, but it could be possible that the deregulation of ROA pathway could be also involved, but I don't know, sorry. Then mouse model. Paul Saultier, thanks Hana for the talk. In mouse models of targeted new variant for transcription factors, the mice carrying a heterozygous deletion of the transcription factor frequently have Y-type phenotype. This contrasts with what is observed in humans. Heterozygous cases have thrombocytopenia. What is your opinion on that? So I don't know about which transcription factors do you think? If it is RANKS1 or not? Probably maybe what is different is the deletion. The heterozygous deletion is not always, it's only the haploinsufficiency like, and maybe in this case, we will not see the same phenotype as with heterozygous mutations that can acquire maybe they can induce other phenotype or acquire other function. I don't know if this is the question. And then is the monoallelic step during megakaryopoiesis described for other genes? Are circulating platelets exclusively normal platelets? And however, an increase in MHEMO is observed in circulating platelets. So monoallelic step during megakaryopoiesis, we have described this only for FLI1. We didn't see it for other genes that we have studied, but we have studied only a few of them, not a lot. So I cannot conclude about that. I don't know. I'm not sure. For circulating platelets, are circulating platelets exclusively normal platelets? However, I don't see an increase. I mentioned is observed in circulating platelets. We have observed the MIH10 increase in circulating platelets, and if they are, yes, I think that. So the question probably is whether the megakaryocytes which are not forming, which are going to apoptosis, probably this is the question for FLI1. Yes, now I understand. So if the defective megakaryocytes are going to apoptosis, I think that Marie-Christine think that they are not able to generate platelets, and the question is why we observe MIH10 in the circulating platelets. So this is a good question. So that means that we have also a defect in the megakaryocytes which are generated. So probably the level of FLI1 is also affected. It's not only a question of monoallelic and biallelic expression. I agree with that. Thank you. What do you think are the best available techniques to capture the complex interplays between transcription factors, which are frequently involved in large transcriptional complex? Good question. Capture the complex interplays. Oh, I don't know if I well understand this question. I'm not sure. No, I don't know. Yeah, I don't know. I am not sure that I understand the question. Which are the best available techniques to capture the complex interplays between transcription factors? Yes, probably we will have, because this the transcription factors are not regulating the same genes. So you have in this complex you have RANKS1, FLI1, GATA1, TAL1, GATA2, and sometimes they are, on some genes there is the cooperation only of on two of them. On some genes there is a cooperation of 3 of them. So this is not easy to answer. Maybe the transcriptional analysis would be helpful to show which targets are common between, for example, RANKS1 and FLI1, or RANKS1 and GATA1, and so on. Okay, so I see no more questions. Yotis, perhaps I miss it, but what proportion of thrombocytopenias are caused by mutation? You focus on, conversely, what proportion are undefined? I think that there is still a lot of thrombocytes. 50% probably of thrombocytopenias are not yet defined. So the proportion is probably half, only half. But the works are, I think that Kathleen Frezon is the good person to answer this question, because she's studying a big cohort. And there is a lot of new genes. Between them, there is also SLFN14, as Marie-Christine Alessi is asking about that, and this was very recently described, SLFN14, and Kathleen Frezon is also working on that. So she could answer this question. I never studied that, so it seems that it is involved in the ribosome biogenesis, but I think this will depend on the type of mutation, and we should do the knockdown to know what is the true role of SLFN14. So yes, there is a lot of things to discover. Yeah, so I don't know if there is other question. Maybe I missed something. No, it seems that's all.
Yes, I don't know if we can wait 1 minute just to see if somebody wants to write the questions in the chat.
So
No
more questions.
No, so thank you so much, Hana, and thank you so much, everybody, for attending today. And do not forget to join us the next session that we will be held the 11th of October. And thank you so much. And thank you so much, Hana, one more time for this nice presentation.
Thank you very much. Thank you.